Welcome to the Explainer. Today, we're jumping right into the molecular balancing act. We're going to completely map out the incredibly precise biochemical control systems of human metabolism. Specifically, we'll be tracing the intricate pathways of three critical hormones: insulin, glucagon, and adrenaline. We'll follow them from their synthesis all the way down to the exact molecular cascades they trigger inside our cells. Let's get started. Okay, here's our roadmap for today. We'll start with an introduction to cell signaling, then move into understanding hormone mechanics. After that, we've got insulin synthesis and action, diabetes and insulin resistance, glucagon and the hypoglycemic response, and finally, adrenaline and signal amplification. Let's dive in. Section 1. Introduction to cell signaling. Any organism made up of more than one kind of cell faces a fundamental challenge. It has to communicate. It has to transmit signals internally and sense what's happening outside. To pull this off, the human body uses three main chemical signaling systems. We have the nervous system, the immune system, and, importantly for us today, the endocrine system. We're going to focus exclusively on these hormonal signals. This is the exact system dictating how your body handles the sugar from your morning coffee or the stress of an unexpected deadline. Section 2. Understanding hormone mechanics. Before we map the pathways, what exactly is a hormone? The word itself comes from a Greek root meaning to stimulate. In biochemistry, they're active chemical substances that interact with target cells to produce a specific response. And what makes them so genuinely fascinating is their efficiency. They operate at astonishingly low concentrations, thanks to a biological phenomenon called signal amplification, which we'll map out shortly. But here's the golden rule of endocrinology. A cell is only a target if it actually holds the specific receptor key for that exact hormone. If a cell lacks the receptor, the hormone just floats right on by without doing a thing. Now, hormones can travel far through the bloodstream, but they can also act locally. You've got autocrine hormones that act on the exact same cell that fired them, and paracrine hormones whispering to their direct neighbors. In all cases, though, the receptor is the master key.
Where that lock, the receptor, is located depends entirely on the hormone's chemistry. Take peptidic or amine hormones like insulin and glucagon. They love water, meaning they can't slip through the lipid barrier of a cell membrane. So their receptors have to sit right on the outside of the plasma membrane, triggering a second messenger inside the cell. But steroid hormones? They're derived from cholesterol. They're hydrophobic. They just slip right through that plasma membrane and head straight for the nucleus to alter gene transcription directly. Section 3. Insulin, synthesis, and action. Let's look at our body's ultimate builder hormone, insulin. It's synthesized specifically by the beta cells of the pancreas, but not in its final active form. It actually starts as a larger precursor called pre-proinsulin. First, a limited proteolysis chops off the signal sequence, turning it into proinsulin. Then, another cleavage removes the connecting C-peptide. What you're left with is mature insulin, an A chain and a B chain locked tightly together by stabilizing disulfide bridges. It's a highly rational system, really, keeping insulin stored in an inactive state until there's an actual spike in blood glucose. So how does the beta cell know exactly when to release it? It's an exhaustive, elegant mechanism. Glucose enters the beta cell through a transporter called GLUT2. Think of GLUT2 as a sensor that only activates when your blood glucose is genuinely high. That glucose undergoes glycolysis, producing a massive spike in cellular ATP. This rising ATP literally binds to and closes a potassium channel on the cell membrane. With potassium trapped inside, the cell membrane depolarizes. That voltage change pops open a calcium channel, calcium rushes in, and that influx signals the intracellular granules to dump mature insulin and the cleaved C-peptide straight into the bloodstream. Fun fact, because they're released in a strict 1-to-1 ratio, checking C-peptide levels is exactly how labs evaluate a patient's natural pancreatic function.
Now, when that insulin actually reaches a target cell, things get really interesting. The insulin receptor is a dimeric protein spanning the membrane. When insulin binds to the outside, it forces a conformational change that activates the inside portion, allowing it to phosphorylate its own tyrosine residues. This is called autophosphorylation. It essentially turns itself on, priming it to activate a cascade of other internal cellular proteins.
Once autophosphorylated, the receptor kicks off multiple complex cascades. The first is the mitogenic pathway. The receptor interacts with insulin receptor substrate 1, or IRS1. IRS1 gets phosphorylated, starting a chain reaction that pushes a transcription factor called ERK straight into the cell's nucleus. Once there, ERK acts like a master switch, activating specific genes for lipid biosynthesis and glycolysis. The cell is literally physically rebuilding itself to process the new influx of glucose.
Then you have the metabolic pathway. IRS1 is phosphorylated again, but this time it activates protein kinase B, or PKB, via a membrane phospholipid called PIP. PKB is crucial. First, it inactivates an enzyme called GSK3, basically ensuring your body keeps storing incoming glucose as glycogen. Second, PKB commands intracellular vesicles containing GLUT4 transporters to physically move and fuse with the cell's outer membrane. Your muscle and fat cells literally open up extra doors to rapidly absorb the excess sugar right out of your bloodstream.
Section 4. Diabetes and insulin resistance. But what happens when this perfectly balanced system breaks down? We get diabetes. We can neatly divide this into two categories based on exactly where the molecular failure happens. Type 1 diabetes is an absolute lack of insulin synthesis. The beta cells just aren't secreting the hormone, so glucose stays trapped in the blood. Type 2 diabetes, which is about 90% of cases, is a condition of insulin resistance. The insulin is there, but the target cells just don't respond properly. In both scenarios, since the cells can't absorb carbohydrates, the body desperately pivots to burning fatty acids instead, leading to dangerous metabolic imbalances. Looking at type 2 diabetes on a biochemical level reveals a very specific list of potential defects. We can divide these into two origins. First, abnormalities in the beta cell's secretion product itself. Maybe a genetic defect causes abnormal insulin molecules or an incomplete proinsulin conversion. The key fits, but it's a terrible fit. The second issue is target tissue abnormalities. A cell might just produce fewer insulin receptors, or there's a post-receptorial defect. That means the insulin binds perfectly, but the internal cascade, all that IRS1, PKB, and GLUT4 stuff we just mapped out, is broken. This is exactly why mapping these biochemical pathways matters so much. Fixing a post-receptorial defect requires totally different pharmacology than treating a simple lack of insulin synthesis. Section 5. Glucagon, the hypoglycemic response.
Now let's meet insulin's direct antagonist, glucagon. This is the great mobilizer hormone, secreted by the alpha cells of the pancreas when your blood glucose drops too low. Just like insulin, it's a peptide hormone, so it can't enter the cell. Instead, it binds to a 7-helix transmembrane receptor, or GPCR. This receptor snakes back and forth through the cell membrane 7 times, and on the inside, it's coupled with a trimeric G protein made of alpha, beta, and gamma subunits. When glucagon binds to the outside, the receptor shifts, forcing that alpha subunit to physically separate from the other two. Watch how intricately this cascade builds. That newly liberated G-alpha subunit slides along the inside of the membrane until it docks with an enzyme called adenylate cyclase. Adenylate cyclase wakes up and starts wildly converting cellular ATP into cyclic AMP, or CAMP. This CAMP is your vital second messenger. It travels through the cytosol, binds to and activates protein kinase A, or PKA. PKA then goes to work running throughout the cell phosphorylating target metabolic enzymes. It phosphorylates glycogen phosphorylase to start chopping up stored glycogen into free glucose, and it phosphorylates acetyl-CoA carboxylase to halt lipid synthesis. Literally every action here perfectly opposes the storage commands of insulin.
Section 6. Adrenaline and signal amplification.
Let's round out our metabolic map with adrenaline. Interestingly, adrenaline uses the exact same GPCR cellular mechanism we just saw with glucagon, but it's triggered by acute stress, like a sudden jump scare, rather than just low blood sugar, and it acts mostly on muscle tissue. Adrenaline is an amino acid-derived hormone synthesized in neurons and the adrenal medulla. It starts with tyrosine. An enzyme called tyrosine hydroxylase, which is the crucial rate-limiting step here, turns that tyrosine into dopamine. Another quick enzymatic step converts that dopamine into adrenaline, ready to flood your bloodstream the second you need to fight or flee. So, how does a tiny drop of adrenaline give you that explosive, immediate energy? It's all about the staggering math of signal amplification. Let's trace it. One single molecule of adrenaline binds to a receptor. That one receptor activates 10 G-alpha molecules. Those 10 stimulate 200 cyclic AMP molecules. Those activate 100 molecules of protein kinase A, which activate 1,000 molecules of phosphorylase kinase, which activate 10,000 molecules of glycogen phosphorylase, which ultimately slice off 100,000 molecules of free blood glucose, a 1 to 100,000 amplification happening in fractions of a second. It's absolutely brilliant. If we step back and look at this whole molecular balancing act, you'll see a perfectly elegant unifying motif. It's essentially a binary switch. Glucagon and adrenaline mobilize energy by forcing enzymes into a phosphorylated state.
Insulin stores energy by forcing them into a dephosphorylated state. They're basically fighting over the exact same molecular levers in your body. And to protect themselves from overstimulation once things normalize, your cells will actually desensitize. They literally pull their surface receptors inside, hiding them away until the balance is completely restored.
We've unraveled a lot of threads here today, but I want to leave you with a thought. If we can map every single microscopic step of these cascades, from IRS1 all the way down to the exact phosphorylation site of a specific enzyme, how entirely will modern biochemistry be able to precisely target these pathways to finally cure complex metabolic diseases? We're no longer just observing symptoms. We are reading and editing the molecular blueprint of human energy itself. Thanks for joining me on this explainer. Keep questioning, and I'll see you next time.